Hello everyone and welcome to day 2 of the Cell Therapy Manufacturing and Gene Therapy Digital Week. I'm your host and the producer for the event, Sarah Linton, and I'll shortly be handing you over to our presenter for the session, Alain Pralong. We have a further 5 webinars to bring you over the next 3 days, covering a whole range of topics, and your registration gives you access to all the sessions. You can find a resources page in the main menu with related white papers and exclusive video interviews. And there is also a forums tab in the menu where you can openly discuss any of the topics from the webinars or the wider industry. We've had a few questions over the timings of the webinars after the clocks changed in Europe this weekend. As advertised, we'll be hosting the first webinar at this time each day. That's 2 p.m. GMT, which is 3 p.m. UK time, 4 p.m. Central European Summer Time, and 10 a.m. Eastern Time in the US. And now to today's presentation with Alain Pralong, Senior Vice President of Manufacturing Operations at Cell Medica. He is speaking on developing a stringent, commercially viable manufacturing strategy, anticipating your endpoint. The PowerPoint presentation will last roughly 40 minutes and will be followed by a Q&A. If you have any questions, whether during the webinar or at the end, please do type them into the box provided below at any point, and we'll try and get through as many as possible at the end of the presentation. Finally, if you have any technical issues, please click the red live support button and someone will be able to help you with any problems. So with that, I pass over to Alain.
Thanks for the nice introduction, and as announced, I would like to talk to you today about developing a stringent, commercially viable manufacturing strategy anticipating your endpoint. In this presentation, I would like to go on presenting what is the current experience with monoclonal antibodies and vaccines. Where do we stand? What can we learn and transpose to the field of cell and gene therapy? What are the challenges that are specific to cellular immune therapy? And then end the webinar with a summary and conclusion. Where do we stand today with MABs and vaccines? So essentially, biopharmaceuticals have a very long history. The history started originally with vaccines, and this in the event of treating and preventing smallpox. Recombinant DNA technology has transformed significantly biopharmaceutical industry since its advent in the mid-70s, and it's an industry that is growing since decades. Interestingly, on the picture you see on this slide, it is taken from the Golden Mirror of Medicine, which have been published in 1742. It shows an early vaccination in which a young boy is injected through this tube with some smallpox in order to get a vaccination through the nose. And when we look today how the whole context of biopharmaceutical industry is in the context of economy, then the sales forecast in 2010 was that 5 out of the top 10 products sold are biopharmaceuticals. And this has dramatically changed, as the sales forecast in 2014 was already 8 out of the top 10 products are biopharmaceuticals. And you see that most of these are monoclonal antibodies, and it shows the big evolution and the big momentum that is in this industry and the importance of biopharmaceutical industry over time. When you look at the evolution going further and you see the repetition of the biopharmaceutical market in 2016, then you see on the left side the business as it was in 2010 with 856 billion. Then there was growth of brands, patent expiration, so it's the patent cliff that becomes real, then the business that is created by generic biosimilar drugs, and then the growth that is really in the emerging markets and others. So the importance for biopharmaceutical industry is really to go into these emerging markets, as there is the big growth potential for going to the future. And when we look where we stand today, then we have big multinational companies that have significant product portfolios. They have also significant global manufacturing infrastructure, and they have significant disease control and therapeutic successes. And associated with all that are significant commercial successes. Just when you look at the pictures of these two sites, on the left side you see Genentech's Vacaville facility and site in the US, and on the right you see GSK facility in Wavre in Belgium. Just to give you a bit the ballpark number, creating this whole site in Wavre has cost more than 5 billion euros that have been invested over the last 20 years by GSK. And also from a technological point of view, there has been quite an evolution. So there has been an adaptation and adoption of embedding of new technologies, a massive evolution of single-use technologies, cost reduction, simplification, safety has been pushed, and closing of the manufacturing process is a very important feature of using these new technologies. And as you can see on the left side, you have the tea flasks that have been used initially and are still in use to do cell cultivation, but there has been a massive evolution in technology, and today you have single-use bioreactors going up to working volumes of 2,000 liters. And it is today possible to embed a complete manufacturing process for a monoclonal antibody or a vaccine to a significant scale in single-use technology end to end. The primer for this evolution is well captured in the quote of Antoine de Saint-Exupéry. When you translate it, it says that perfection is achieved not when there is nothing left that you can add, but when there is nothing left that you can take away. And knowing the context of Saint-Exupéry flying airmail over the South Atlantic in the early 30s, there every kilogram, everything that was not needed was corresponding to precious flight time. In a time where you didn't have satellites and weather forecasts of the accuracy that you have today, so flight time was very important. And the pictures you see below show essentially the systems of installed in a biopharmaceutical plant in 1982. So you have two operators there in a fully analog control based manufacturing facility. You have then in 1994 the first installations of the IT revolutions that take place, and then you see in 2009 a fully autonomous 2,000 liter working volume single-use bioreactor. And closing the manufacturing process, going to single-use technology has also enabled to choose new facility layouts. When you look at all the facility layouts, it's a sequence of independent rooms where people have to go in, have to go out again to go into the next room. And with these new technologies, you can essentially build ballroom concepts in which you can use some devices like the controlled environment modules, which are only encapsulating the equipment that is used to do a certain step to achieve segregation, but not more. The new equipment that has been developed offers also the possibility to go to a next step facility design, which is based on modular construction. Modules can be prepared and then shipped to the facility where they are just assembled on site, and the mobile single-use equipment is wheeled into the facility and can be installed. Both the combination of modular construction and single-use technology has enabled significant reduction reduction of capital expenditures to create a manufacturing facility and operational expenditures savings for operating this facility. From the point of view of the products, when we look at the vaccines, then most of the vaccines are at maturity stage. Given the very long history of vaccines, which besides what the Chinese have done, as I have shown for smallpox in one of the first slides, it was then Edward Jenner that was the first which really has tested vaccination. And with the golden age of vaccines going in the 20th century, then you have today a lot of products which have reached commercial maturity. And when we look at the monoclonal antibodies, then the patent cliff that has been announced and mentioned quite a significant numbers of time is also becoming reality and has already become reality with a big push to biosimilar generic products. Furthermore, what has happened also is that the development cost for a new biological entity has massively increased. And you see their data which stems from 1990 and goes to 2012, and in certain legislations, the cost for developing a new biological entity has practically quadrupled to quintupled. While at the same time it has also become more difficult to find new drugs which are successful. So you have more risk in developing a new biological entity and you have more attrition. This is essentially a consequence that quite obvious targets for these are now already products existing, while it becomes more and more difficult to find new targets which can be effectively treated. So when we look at the biopharmaceutical industry and we look at where monoclonal antibodies and vaccines have come to, then we need to recognize that we are in an evolving business environment. On the left side in this table, you see, well, what are the competitive advantages today? And on the right side, you see what are the basis for competitive advantage in 2020. So when we begin, then go from left to right. So today, well, development, resources, sales, and marketing scale, everything is integrated in the same company. And in the future, is the value of products and services is important, and the distribution strength is important as well. From the pricing point of view, you have global high pricing, which potentially restricts access. And in the future, pricing is based on the ability to pay, driving volume uplift. So essentially, you adapt your pricing to the population you want to sell to. Then when we look at competition, you have today multiple competitors in major therapeutic areas, and scale is enabling success. In the future, you will have fewer competitors in a broader range of diseases. We have today multi-billion-dollar drug revenues covering high fixed costs, and we will have in the future more products with lower revenue and lower cost. We have end-to-end operational capabilities for self-sufficient strategy, and in the future we have significant outsourcing of operations such as manufacturing and support functions. Acquisitions of technologies and products to augment our product pipeline is one of the main strategies today. Well, in the future, it will be greater collaboration with academia, biotech, and peers. And the focus is on mature Western market, and in the future, it will be on emerging markets, as I have already shown previously on the slide, which showed where the growth potential is from an economic point of view. Another point which is important when you look at the future and where we stand today with biopharmaceutical industry is that we are still in the environment of a growing world population. Today we are around 7.4 billion people, and it is forecasted that by 2050 we will be around 10 billion people. And you can see in this picture, in this graph, where the growth takes place. Interestingly, in Europe there will be a decline or a stabilization of the population. In parallel to this, there is also a change of the demographics, and this shows that we will go more to a column form in the demography than this pyramidal form. So essentially we will have practically as many young people as we will have elderly. And still today, a big problem is access to drugs, and this is still not ensured to all, and that's the reason why we have UNICEF, why we have PAO, why we have Gavi and all these associations. Because for certain people, when you look at how much money they have per day to live, the drugs that we produce are simply inaccessible. So the question is where we are today and how mature is the biopharmaceutical industry? Are we at the flowering stage? Are we at the growth stage? Or are we already at the harvest stage? So for me personally, this depends at what aspect of biopharmaceutical industry you are looking at. In certain areas, we are for sure at the flowering stage. At others, we are at the harvest stage. But at most, we are probably still at the growth stage. So a lot of things are still in the making, are being developed, and this despite the big economic success that biopharmaceutical industry has. Why do I make this claim that we are still in the developing stage? Well, when you look at the current biological product shortages, there is recurrently a shortage of certain products. It's not always the same, but it's a thing that is recurrent. Most interestingly, it's mainly vaccines that are have this problem with product shortages, which is for sure associated with the complexity of vaccine manufacturing. And the consequences of product shortages can be dramatic, as it is the case, for example, in tuberculosis, when people have to stop their treatment because there is a product shortage or there is an issue with accessibility. Even if they can get the drug later on again, it will not have the same efficacy because there is even a risk that the resistances are created by these microorganisms while the treatment is not followed in the most optimal way possible. A further issue is also the regulatory oversight. So in 2012, the FDA has issued 787 for 183 forms during their inspections. And when you do the ranking of what are the most prominent findings, then you find that 6 out of the 15 most mentioned or cited rankings have to do with following procedures. So essentially with personnel manufacturing drugs being able to follow procedures, working according procedures, or just preparing procedures or being trained in the correct way to be prepared to follow these procedures. So there is a big need in the industry to make sure that we have robust manufacturing ways of working that enable our personnel to follow procedures in a way that gives them the possibility to manufacture these products with the best quality possible. And another issue that we face is that we have primarily a reactive way of working. We have a problem, we see that the problem is there and needs a resolution, and then we will begin to resolve the problem. But it's not really a proactive way. First, we need to recognize the problem. It's much less proactive risk mitigation / risk prevention. However, we have developed a very good capability for crisis management. When we have a problem and the problem boils up, we are not short of means that can be invested to resolve these issues and get them resolved as efficiently as possible. However, working in this way is very resource intensive, it is time consuming, and it is a major risk exposure for the company. What are you doing if the problem cannot be resolved in time, if manufacturing needs to be stopped? So one of the risks is that the consequence is a product shortage, as we have seen before. How can we resolve these things? How can we go about it? And Jason Fried is a very interesting person because he's a co-founder and CEO of a company which is called 37 Signals. And he is able, with a very small company, to compete with much larger companies like Microsoft and others. But it's a quote which he has published in his book Rework. A complex system designed from scratch never works and cannot be patched up to make it work. You have to start over, beginning with a working simple system. But with crisis management and trying to resolve crises once they happen, even by investing a significant amount of means, is trying to patch up a system which is not working to the level it should. So we have to think about how to resolve this thing and come to another way. So we need to understand that product development, and I'm pretty sure that's also your experience, is a bumpy journey. And in that sense, we have to see what we can take over from other companies. How do other companies manage product development and product manufacturing? And an interesting company is Toyota, which is one of the founders of the Toyota production system. And you see here the house of the Toyota production system, which is just in time, it's automation, it's load leveling, it's it's a 5S, it's 7 wastes, suggestion systems, defect warning, total preventive maintenance, standard operations. And in that framework, Toyota tries to bring the best quality to the customers by having a high quality production system. Interestingly, they have per year more than 700,000 improvement suggestions from employees working at Toyota at all levels. And that is an average of more than 10 improvement suggestions per employee per year. And they are able, with the ways of working they have established, to embed over 99% of the suggestions and to implement these. My personal experience is that at one company I was working, one of my bonus relevant goals was to come with one improvement suggestion per year. And this shows already quite a difference where Toyota is in their approach and their flexibility in reviewing the way how they do things and the way we normally do in biopharmaceutical industry. Well, how do they do it? And the The answer is relatively simple. They embed operational excellence. They have understood that multiple actors, QA, regulatory affairs, QC, operations, research and development, need to collaborate and they have to work together. So multiple activities in these different departments need to be executed in parallel. And these activities need to converge at specific moments in time in the development of a product or the manufacturing of a product and for sure its release. So it's creating a powerful union between Lean and Six Sigma. Lean is about speed, getting momentum, and Six Sigma is about stability and accuracy. But both together are focusing on customer satisfaction, employee growth, profitability, and increased volumes. And when you look at that in product development, then on the left side we have a bit the way how product development is often done today. So you have a bunch of birds flying a bit in all directions, and in that case you have questions that you need to answer is how can we get better view from discovery to the launch of a product? Where are my raw material suppliers? What are the constraints of manufacturing? What is the impact of my job on the launch? How can we get more collaborative relationships? Are we aligned internally? Do we have the right talent? How to ensure that we understand what brings value? And once you are applying these questions and trying to answer these questions in the context of developing a product, then the objective must be to bring clarity and alignment. And one tool to do so is a tool like the product development value stream, which is essentially giving a clear direction to the organization how to go forward and how to work off the projects. So the development pathway is structured into stage gates in the development value stream. And this is a tool that has been developed in the frame of two positions that I held at companies. And the idea was really to structure the development process so that data can be gathered in time to make science and data and fact-based decisions. And with the stage-gated process, you have essentially 6 major stage gates. One is commit to research. Then it's commit to candidate development, commit to phase 1, 2, go to final scale, commit to phase 3, and commit to file and launch. And to pass each of the stage gates, you have to have compiled a significant number of data and results that are specific for this stage gate. What needs also to be understood is that product development takes place in a circle which is combined with technical lifecycle management. At the beginning, you develop a product, you develop it in the best case to the launch, successful launch, you begin manufacturing, and with the manufacturing starts the technical lifecycle management because we live in an environment that is changing. Suppliers change, raw material suppliers change, other goods can change. You might need more capacity, so facilities change, and in that sense we have to maintain the product and its process over time. And that's technical lifecycle management. And the purpose and scope of lifecycle management in general is the modulation of the product lifecycle. At the first, you have a big investment, so that's the R&D, which is on the left side. Then you come to the launch and you develop the product to maturity, and then over time the product evolves in the life cycle and goes down. And trying to modulate this is one of the reasons why life cycle management is done. So it's getting most of the out of the product that has been developed over time. And another tool that has been developed to manage the technical life cycle is the technical life cycle value stream. As I outlined earlier, we are currently working in a reactive mode of action. We have a problem, the problem often boils up to an issue, maybe even to a crisis, and then we will react. One more efficient way of working would be to try to recognize and to anticipate what might be problems of the future, what might be issues, what are our risk exposures, and then to preventively begin to work on it. And for this, the technical lifecycle value stream has been developed, which is essentially a stage-gated continuous improvement program. So from a proactive point of view, you do first a diagnose phase where you look at your products and processes and make a risk assessment. This risk assessment is rated through a coherent system of questions that you ask, so to make sure that all risks are assessed with the same scale. Out of that, then the biggest risk exposures can be isolated, can be recognized, and can then be treated in the design phase, where essentially programs are developed to resolve these risks and reduce the overall risk exposure. And that will require changes, and these changes are then implemented in the implementation of changes phase. And when you combine the product development value stream and the technical lifecycle value stream, then you have a holistic system that ensures quality in developing products and maintaining products. So you see, first, quality is built in because you have clear Toyota principle-based systems. You have all the embedding of Lean Six Sigma principles, so quality is built in. You have also a life cycle approach from development to product discontinuation. There is also an understanding of the complex supply chain and the CMO networks. There is a robust process measurements and analytical tools. There is real-time assessment of product and process capability. There is maintaining of the state of control throughout the commercial life cycle, and there is ensuring an effective and timely change management system for continuous improvement and optimization. What this slide shows is that with the development of the product and the finalization of its development, a big other part begins, and that's the whole technical life cycle value management. And this should be done as much as possible in a proactive way in order to protect the company from the risks that can accumulate over time in a process and a product that is not maintained. When you look at this picture of the kids there, probably these kids are 5, 6.5, 7 years old. When you look at the plan of the set that they should build, they are probably to build this set with 5.8, 5.9 sigma accuracy. So they achieve a very high success rate and a very high quality. When we traditionally would work in biopharmaceutical industry to build this set, we would probably prepare standard operating procedures of 25 pages, and we would achieve 2 to 3 Six Sigma. And that shows essentially, and leads me back to the findings of the FDA in their 483s, that documentation, training of people is a huge issue in our industry, which needs to be absolutely tackled. And we have to find ways that enable us to train the people correctly and really get the people where they are. It needs to be adapted to their capabilities and it needs to be adapted to their work environment. That will be very important going forward, and there had been several initiatives in the past and are also going and have been published a few months ago, which described quite well what the issues are and how these things could potentially be tackled. Now, after all this information and the status on where we stand with monoclonal antibodies and vaccines, which are essentially established multi-billion revenue generating industries, and we have seen where they are strong, what are a bit the missing links, and the whole industry is still being matured and is ripening as we go, and in the whole frame of already delivering very successful drugs to patients, the questions will come: what are the challenges for a relatively novel industry like immune therapy? So one of the big challenges is simply that cellular immune therapies are personalized medicines, or better said, personal medicines. It's a new class of products, and there is not a lot of experience in regulatory affairs how to handle this. There is a high interpatient variability with the starting material. Compared to vaccines or monoclonal antibodies, we are not working with master cell banks and working cell bank systems, master virus and working virus systems that are well characterized and validated. We start with the blood sample from a patient, and there is no stockpiling possible. So if monoclonal antibodies manufactured, they have big quantities on stock. With vaccines, they try also to have big quantities on stock. As it is a personal product and it is manufactured for each patient individually, there is no stockpiling possible. And this gives together a significant risk to the supply chain. So complex manufacturing protocols, semi-open aseptic processes. As everything is low volume, a lot of these manufacturing operations take place with lab equipment, and it's not closed or fully integrated. It's manual, it's operator-dependent handling. There is currently only limited automation available, and the transport from and to the patient has proven to be complex and complicated as well, depending if our blood or apheresis or so is shipped. And the manufacturing cost per dose is today, given all the complexity and the significant number of operators that are used to manufacture such a personal product, are still high and can limit accessibility significantly. So in summary, monoclonal antibodies are very well established class of products. Vaccines are established as well, but they exhibit issues with the technical life cycle management. That's a consequence of the long history, and that a significant number of these products have been developed in the 50s, 60s, 70s, and are still manufactured today in the same way. The main focus of monoclonal antibodies and vaccines is today on accessibility. And monoclonal antibodies and vaccines have extensive experience with regulatory affairs. And just the fact that monoclonal antibodies are considered as well-characterized biologics shows that regulatory affairs, quality control, QA, and all these functions are very well settled in maintaining the product and the process in a state of control for monoclonal antibody manufacturing. What I try to convey as well is that structured approaches exist for product development and product life cycle. It can be done. And cellular immune therapy as a new class of products offers a massive promise, especially in oncology, where a number of companies have shown spectacular results recovering people in the final stage of the disease and showing the potent therapy these immune therapies offer. And it's a young field in manufacturing. It requires a steep learning process as a consequence of its novelty and its short history, and to make these drugs available to the biggest number of people in need. In conclusion, the field of immune therapy and cell and gene therapy can build on the experience of monoclonal antibodies and vaccines. So to do so, there is required to embed a structured product development process. And this is about control. Control your pipeline, control its development, control the costs, and control the associated risks. It's about time to market. If there is not a structured product development process, time to market is at risk. It's also to reduce the regulatory hurdles, because you fill in the data as you go and you fill it into the level that the regulatory authorities can accept the data that is generated. And all the knowledge and the structure that is built in will enable life cycle management in the future. But to be successful in embedding such a product development process and to have this structured, it requires a holistic approach. And the holistic approach begins with humans, because it's humans that are working in the facilities, it's humans that are developing these products. Then it is about products, so the processes and the analytical methods. Do we understand the products? Then it comes to technology and facilities. What is required to manufacture recurrently a product of the desired quality? And then it comes to timelines, which is also a focus. And the whole thing needs to be managed in a holistic approach, where these different elements and parameters are diligently balanced. Thanks, and I'm ready to take your questions.
Thank you very much for such a fascinating talk there, Alain. Just a reminder to our audience that now is the point where you can ask questions to Alain that you may have. We do have one question here. What are the main differences in the maturity of the monoclonal antibody or vaccines manufacturing compared to that of the cell and gene therapy manufacturing, Alain?
Well, the main difference is that with monoclonal antibodies, there is now a history that is 30 to 40 years long. With vaccines, it is even much longer. And with cell and gene therapy in the manufacturing space, it is relatively new. And in that sense, we witnessed the same growth pains as we had at the beginning when monoclonal antibody manufacturing or vaccine manufacturing was set up. So it's a matter of time until the critical elements and the risks that are associated with this manufacturing are fully grasped, so that the technologies can be incepted and developed on one side, and the economics of manufacturing tweaked and optimized on the other side. And on the third line, not to forget, is to also collaborate with the regulators, that the understanding is growing what the specifics of this new industry as part within the biopharmaceutical space is.
Okay, fantastic. And there's another question here. One topic that's been coming up a lot is automation, and how people working in the cell and gene therapy space could probably learn a lot from, the automated systems that are already being used when working in, monoclonal antibodies. What are your thoughts on that, and what do you think the specific lessons that they could learn could be?
Well, automation is for sure something that is absolutely required in order to make manufacturing feasible for these class of products. And given the fact that we do not have the traditional means to do economy of scale as we see with MAB or vaccine manufacturing, where when you double the size of the bioreactor, you can expect practically the double output. That's not the case. So it's much more a manufacturing model which needs to answer the question, how many products can I have in a throughput per unit of time than in scale? And that is a way of thinking which is well embedded in other industries. Just when you think about car manufacturing, or when you think about manufacturing technologies where it's the piece number that counts, and we are until now much more a volume-driven manufacturing industry, volume in the sense of capacity in liters in bioreactors, and this model is changing with this class of products.
Perfect, thank you. We have another question here. What process controls need to be in place for cell therapies that were not needed for monoclonal antibodies and vaccines?
Can you say again the question, please?
Of course. What process controls need to be in place for cell therapies that were not needed for monoclonal antibodies and vaccines?
Well, I think the main difference of the control is the release assay and the whole supply chain management. Until now, when you were manufacturing a monoclonal antibody, you could do that by yourself, and you could stockpile, and you can release the product. And even if you had a batch which had some deviations and all that, you would keep that normally in stock until you have really to scrap it and in the best case, you can resolve deviation. The main difference with cell therapies is that stockpiling is not possible, except for maybe allogenic products in the future. But for that, it's a product comes in, the material has to come to the factory in the best state possible. So there needs to be control of sampling of the patient, isolation of this material, and shipment of this material to the factory on one side. Then the product needs to be manufactured, and it needs to be released within a period of time which is meaningful for the patient, meaning that the product needs to go back to the patient as fast as possible. And then it should come in a good state to the patient and should be also treated and handled at the hospital, at the healthcare institution, in a way that the product is active when it is infused in the patient. So there are a lot of things which controls need to be built up, which are not seen traditionally in the manufacturing of monoclonal antibodies. But, however, from a process perspective, well, you need to control pH, you need to control the environment, you need to control feeding and all this. So in that sense, there is not a massive difference. One thing that is a big question that will need to be answered is that some of these processes use again serum, and with the monoclonal antibody world, we were nicely going away from these kind of components.
Fantastic, thank you, Alain. We've now covered all the questions that have come in, actually, so I think we'll wrap the session up here. Our next session of the week's tomorrow at 2 p.m. GMT, which is 3 p.m. UK time, 4 p.m. mainland European time, and 10 a.m. Eastern time in the US. And tomorrow we have two presentations. The first is coming from Professor Mark Liddell, who's speaking on planning for success, preclinical and phase 1 trial considerations to prevent failure at marketing authorization approval. And then our second talk will be coming from Janneke Mullenberg, who's from Vicinivax, and she'll be speaking on clinical development of VVAX 001, which is a Semlinki forest virus-based cancer vaccine for the treatment of HPV-related pre-malignancies. So with that, I'd like to thank Alain and thank everyone for listening, and we look forward to welcome you to Digital Week tomorrow. Thank you.
Bye-bye.